So, yeah, so the title of my talk is to highlight the importance of the within species biodiversity and how that is relevant to management. And the focus will be on Scandinavia and particularly on the sea outside here and outside of Bohuslän in the Skagerrak, with some important lessons also about how to think about climate change effects in the future. So I was asked to include also some of the Sustainable Development Goals in the presentation. Whoops. And these are the ones I think that you can think about. So in the bottom right, I also included the Biological Diversity Convention, which is really important. So why is it important? First of all, what is biodiversity? So biodiversity encompasses everything from the smallest level of DNA sequences to big ecosystem effects. But what I will be talking about today is sort of a sometimes forgotten boundary or level here, which we call the population, which lies somewhere in between individuals and species, and it can be defined as a sort of functional unit, which is a group of individuals that actually meet and interbreed freely in nature and is then more or less connected to other nearby populations within a species. So why is it important to conserve biodiversity? Well, there's many reasons. There's a lot of biological resources in nature, a lot of compounds that we can use for medicines. There's diseases that can come from nature that we need to know about. There's also functions provided by nature to us. And also, in order to conserve nature and restore, we need to know about biodiversity. And that is sort of the focus of today's talk. So, knowledge about where you have boundaries between different stocks or different populations, I'm using these two words interchangeably, is really important in management. So one really good example that you have here is on the Atlantic cod, which is known to have two stocks, one in the North Sea and one in the Baltic Sea. And knowing where the boundaries are between those stocks is really important if you want to manage these stocks and regulate the fishery. It's also important if you want to protect the species by putting in protected areas there. Where do you place those? And also if you want to restore a damaged environment or a damaged population, you need to know about where these boundaries are. So how do these boundaries arise? There's multiple different ways these can arise. It can be due to history. So for example, when the ice age was at its maximum here, we had ice and the sea levels were lower, which meant that populations in the sea were more isolated from each other. As sea level rose after the glacial period, populations could again, or individuals could move freely between different areas, but this historic separation could still have a signal. You can also imagine that environmental gradients, such as the Öresund here, where you have a very strong salinity gradient, can also create a boundary where you can have populations that are adapted to low salinity in here, but adapted to high salinity out in the North Sea. Finally, you can also have barriers due to, for example, current patterns that prevent larvae to spread from one area to another. And so when you have these different stocks, you have potential consequences of that. And one consequence of it is that it can allow for local adaptation. So you can have local stocks that are more tolerant to, for example, future change, which can be good. But you also separate the stocks into smaller parts, where each part is more vulnerable than one large one. So this is important to know about this. And so the processes that create these patterns are twofold. One of them is the lack of migration. So if you imagine here you have one big population that gets separated in space by a barrier, and then these individuals cannot swim from one to another. With time, genetic differences are going to arise randomly, and since they are not mixing here, these random changes are going to create differences between these two populations that are now in different areas. So that means that knowledge about dispersal is very, very important if you want to manage genetic biodiversity. The other factor is then local adaptation that I hinted at already. And here you have some of the work of Kerstin Johannesson, who I've been working with. I can say that the work I present here is by no means all my work. It's work from me and also from colleagues that I've been working with throughout the years. So here you have an example of a snail population that colonizes a coastline, where part of this coastline is small boulders with a lot of crabs, and part of this coastline is just rocks where there are no crabs, but there are a lot of waves. So what happens then, if there is no migration here, or if the selective differences are very strong, you can have evolution of locally adapted snails here that have really high fitness, that's the green bar here, in this area. So they are big and thick, they can survive crab predation very well. Whereas here in this area you have locally adapted snails that are really good at surviving wave action. So they survive well here, but if you move them around, they are not doing so well in the other environment. So fitness differences among the local populations is really a key factor as well to consider in marine conservation. So in management, so here is a little bit more hands-on what you need to think about. So when you manage marine fisheries, you need to consider where you have boundaries between stocks and what the relative stock sizes are. I'm going to talk more about that in a moment. In management, when you create marine protected areas, you need to think about where to place those and how they should look. You also need to consider moving things around. If you do that, what are the effects of that? And what are the effects of potentially invasive species when you move organisms around? And restoration, I'm not going to talk so much about that due to time constraints, but here it's also really important to consider genetic diversity when you restore, because you need to think about where to restore and where you get the individuals from that you use for restoration. And then climate change comes on top of all of this, of course. So in fisheries, say that you have two stocks in your system, one big one with 1 million individuals and one small one with 50,000 individuals. If you don't know that these are two different stocks, you might set a maximum sustainable yield based on that you have 1,050,000 individuals. And if you fish then this area as if it was just one stock, you are going to overfish stock 2 here severely. So in this case, it's really, really important to know that you have two stocks of different sizes here in this area, and they should be managed separately. One good example of two stocks that are mixing in our part of the world is in the cod. So this is work of Kalle André and Simon Henriksson, who you have here in the corner. Where in the Skagerrak we know that there are two stocks. There's a coastal stock that lives primarily in the fjords, which is here illustrated in orange, and then there's a more offshore stock of cod, or population of cod, that is in blue here. And in this work, they went out and they sampled cod in 2019, just newborn cod, really small ones, and they found that, as you expect, you have coastal ones in the fjords, and then offshore you have the offshore ones. But then when they went back again next year and looked at the age 1 group, they could see that a lot of the coastal cod had disappeared. So here's an example then where you have these two stocks that are mixing. Another great example is in Baltic Sea herring. So this is the work of Leif Andersson's group in Stockholm, where you don't really have spatial separation, but more of a temporal separation. So the herring are out offshore most of the year, but they come into the coast to spawn, and some of them come into the coast in the spring, yellow here, and spawn, and some come into the coast in the fall to spawn. And what these guys could show was that the spring spawning herring are genetically different from the autumn spawning herring, even though for most of the year they live together offshore. So how do you manage that as a fisheries? Well, it turns out that the fishery has not been well managed in the past, and as a result the stocks have been declining quite dramatically. And this caused the Baltic Sea Center at Stockholm University to produce this policy brief a while back, where they really stressed to managers that they need to use DNA techniques in order to do surveys, and then use this to include it in management. And I think now, just recently, the EU Commission recommended closing down the fishery completely. So if we move up from one species to multiple species comparisons, what are the trends that we see? Well, one big pattern that we see is that the Öresund seems to be a genetic barrier for many species. So here's a review paper from a few years ago that showed many species of both fish but also other organisms. Here you have the entrance to the Baltic Sea. You see the salinity gradient into the Baltic Sea, and you see also these are genetic differences compared to outside of the Baltic Sea, that at the entrance of the Baltic Sea most organisms show a big genetic leap or a genetic differentiation here. So this is also a big environmental difference here, so perhaps not super surprising. But there are other places as well where you see these multi-species genetic discontinuities. One of these is on the Norwegian southwest tip, where here you show for 3 species. This is recent work from Halvor Knudsen in University of Agder in Norway, showing that in many fish species this southwestern tip seems to be a barrier. So you have 2 different populations on the 2 different sides of the tip. So now I'm done talking about fish. I'm going to talk about mussels instead. So in mussels, the story is quite complicated. So here's a recent paper showing in color the different mussel lineages that we have in the North Atlantic. And there's at least 6 different mussel lineage that exist here in the Atlantic. So different populations, or even different species, if you will, although they do hybridize with each other. In our part of the world, if we zoom in, we see that we also have barriers to gene flow, which can result in different populations, also in the Skagerrak. So here you have Skagerrak, you have Sweden, Norway, Denmark down here. And here I include both blue mussels on the left and oysters on the right. And the red areas here are areas inferred by genetic data as barriers to gene flow. So mussels and oysters both are sedentary as adults, but they move around as larvae with currents. And so how the water moves determines how the larvae, where the larvae end up. And what we see in mussels is that in this fjord area around Tjörn and Orust, there's not a whole lot of dispersal of larvae. We also see a barrier here between sort of Sweden and Norway, and also a barrier along the Norwegian coast. In oysters, we see a similar pattern with also a barrier along the Norwegian coast, but we also see a rather big barrier between the Skagerrak and Kattegat areas, actually. So why do we see these barriers? Well, it turns out that where the organisms are located in the archipelago matters. So this is based on current modeling data by Malin Gustafsson at IVL, who has compared sort of the outer archipelagic populations compared to inner ones. Inner ones would be inside fjords and bays. And if we just look on the left here, this is how many sites that each site is connected to in the outer archipelago versus inner archipelago. And you can see in the inner archipelago, the areas both receive larvae from other sites and donate larvae to other sites, much less than in the outer archipelago. On the right here, we see instead local retention. So that's how much of the larvae produced stay where they are from. And you can see in the outer archipelago, it's almost 0. So all the larvae go somewhere else. Whereas in the inner archipelago, a lot of the larvae stay where they came from. If we instead now compare with another species that do not have larvae that swim around, and here we use the isopod, the Baltic Sea isopod, Idotea baltica, as an example. So these bear live youngs, they brood them, they carry them on their bellies and release them as little babies So there's no swimming with the currents very much. Here we see a much stronger structure. So in this area here on the left, we see that there are at least 10 different populations in Scandinavia. So we have one in Skagerrak, one in Kattegat, and then at Öresund is another separation to one. The rightmost figure here is connectivity data based on current patterns as well. And as you can see, the current data matches the genetic data we see quite strongly, because these are not quite strong swimmers, so they tend to just swim around in the algae. What does this mean then, that we have genetic differences between, for example, Kattegat and southern Skåne? Well, we went out and tested that. Do these genetic differences actually mean anything? So we sampled individuals from Vejbystrand here and Ystad in the south, and we compared their physiology. And what we could see when we transplanted them, so we took individuals from low salinity area here and put it in high and low salinities, and did the same thing for the high salinity population here. And what we found was that in the high salinity population from here, there were no real physiological differences when you put them in different salinities. They didn't really care about it in terms of how much they ate up here and how much they respired, how much oxygen they used down here. But the low salinity population from southern Skåne, we saw that when we put that in a higher salinity than they were used to, they ate less and they respired more than they did in their native salinity. So that's a sign that they're not really doing well, right? They use more energy, but they intake less energy. So there are physiological differences here. Another example, if we move away from animals, here is the work of Marlene Janke and others on eelgrass. Here we see a genetic differentiation that's slightly different, that we have 3 different groups, one in green here up in the north, one in blue in the Kattegat, and also we have a red genetic group that's just around the Tjörn and Orust area. So these are 3 different genetic groups. And interestingly, of course, there's a lot of restoration efforts that I'm sure many of you know about, focusing on this area here, which makes it really important to know that this is a evolutionarily distinct lineage that lives here. Does it matter for the eelgrass that the genetics are different? Well, it turns out that yes, also in eelgrass the genetics matter. So here's a recent study by Duffy et al. Where they looked at genetics compared to physiology, and they saw strong correlations. So here on the x-axis all over is genetics, which is sort of a summary here, a multidimensional summary of all the genetic variation in the eelgrass, compared to physiology. And on this side here, on the y-axis, we have how tall are the leaves. So high up here is very tall leaves, and down here low is short leaves. And you can see that the leaf length and genetics correlate quite nicely, both in the Atlantic in blue and Pacific in green. Same thing when it comes to bushiness. So this is how close the plants grow to each other. Down here is very bushy, down here is sparse, that they grow far away from each other. And we can see here again that both in the Atlantic and in the Pacific, the genetics correlate well with bushiness. So the genetics matter for eelgrass physiology. Now, what about the management? So what do you think about with when you try to manage these things, when you have this genetic information? And I want to put this in the context now of the Kunming-Montreal agreement, where we now have targets to protect and manage 30% of areas, including marine areas, by 2030, and also to restore degraded ecosystems by 2030. How do we go about doing that? And we need to put protected areas out there. Here is a map of all the protected areas in Scandinavian waters from the Marine Protection Atlas. But as you can see, all these protection areas are in green, which means that they're not really protected. They're protected on paper, but in order to be really protected, for example, the fishery needs to be limited. There needs to be some kind of actual hands-on limitation, which is not the case in many of these areas. So if we now go to the marine protected area network design, when you design marine protected areas, there's these three things at least you need to consider. They need to protect biodiversity, they should provide connectivity, so it should be possible for organisms to move from one to another, and they should protect suitable habitat under climate change scenarios. So here is a really interesting recent paper where these authors introduce a new framework for looking at how to incorporate connectivity estimates into where you design protected areas. So if we look just in the Baltic Sea network, does it protect biodiversity? If we compare then biodiversity, genetic biodiversity, inside and outside of protected areas, we see that in most species there's no difference in biodiversity here. So these are studies by a review paper from 2017, and then I have included also the Baltic Sea isopods here. We don't see a difference in biodiversity inside and outside of protected areas. For connectivity designs, here's again the same framework that I mentioned before, where you can compare where current MPAs are, which is in green and blue here, and the red dots are areas where they should be placed in order to provide good connectivity. Finally, what about climate change? So here was a recent paper that came out last year that looked at the future effects of climate change impacts. It's a cumulative impact assessment, and what they could see, if you plot it on a map here, is the increase in impact predicted by the end of the century in Swedish waters. You can see in our area here, it's not as bad as in the Bothnian Sea or in the Gulf of Bothnia, but still the impact is predicted to be around doubled by the end of the century. And a lot of this here you see in A and B are due to warming effects or salinity change effects. So what are these effects that we expect to see? Well, one thing is that we expect to see species distribution differences. So here's an example where we have combined, or these authors have combined, current distributions and experiments on tolerances to future salinities and temperature in order to predict what the distributions will look like in the future. And this is for bladderwrack, Fucus vesiculosus on the left here, and for Baltic Sea isopods on the right. And as you can see here, yellow and red means that there's a high probability of finding these species here, and in the future the blue means it's a very low probability of finding these in the future here. Now, if you just look at the distribution, you can gain an understanding of how it will look like in the future, but you also need to include connectivity in the climate change impact assessments. So in bladderwrack, for example, you can do a niche model and you can see that in the future we think that there will be suitable habitat here, but if you include the fact that the fucus cannot disperse very well, you rather arrive at a map that looks like this. So you have much less potential space for it in the future. Finally, I want to mention that evolution also occurs. So things can adapt and change in the future as a response to climate change. And there are currently a lot of new methods ongoing to try to model how this can happen in the future. And there's this concept of genomic offset or genomic vulnerability that tries to predict then how much evolutionary change will be necessary to track changes in the future. And that's based on the current distribution of genetics and the environments, and then you use that with AI methods to model what will happen in the future. And here's an example of a recent study looking at seaweeds in Australia and Tasmania, and they can model then on a local scale how much genetic change is required in order to track global warming. So now, it is this important within species diversity. Why is it not really used so much in conservation, hands-on conservation? It is mentioned a lot in large-scale documents on the global or regional scales, but not so much in national or local scale documents. So there was a recent study by Linda Leikre and co-authors where they tried to look in different documents how much genetic biodiversity is mentioned. And this was their conclusion. So why is this the case? Well, you can imagine a few things. It could be that species-specific patterns preclude generalizations. So there's a lot of differences depending on if you're an oyster or a fish or an isopod or eelgrass. And also that usually the genetic data is quite complicated. So that's a lesson to us, the researchers, that we need to communicate the results in a way that makes sense to managers. So we need to communicate better, and we need to communicate on a regular basis. There was another recent study by Carina Lundmark and co-authors where they looked at the effects of communication about genetic diversity, and they could see that this was a self-ranked by managers how important is genetic diversity before being given a lecture about the topic, and also after the lecture. And you could see that right after the lecture, managers thought that biodiversity was much more important than they thought before the lecture. But 3 to 4 months later, when asked again in a questionnaire, it was back to the levels that it was before the lecture. So they forgot how important it was. So in order to maintain a momentum here that genetic biodiversity is important, you need to communicate often and regularly. So that is the conclusion of this. So to summarize, within species biodiversity is important to think about when you regulate fisheries and also in aquaculture, especially in aquaculture where you tend to move organisms around. It's important for generating marine protected area networks. It's important for restoration, like I hinted at with the eelgrass. And it's also really important when it has to do with predicting the effects of climate change. So hands-on for managers, what is important information you can get from genetic biodiversity? Well, you can get estimates of where population boundaries exist in time and space. It's important to mention here that this is by no means static, right? This changes over time where these boundaries are. If you measure one time, it can be somewhere else next time. So it also highlights the importance of being able to update management plans when things change out in nature. You also get connectivity estimates among geographic areas. So you can think of it as source sink dynamics, or where do larvae come from and where do they go, for example, when it comes to mussels and oysters. And also you can get information about relative adaptive potentials, evolutionary potentials, or vulnerabilities from a genetic perspective across space. And we need to collaborate better between science and policy. So last slide I have here is just to introduce 2 new projects that we have now that try to address this in different ways. One on the left here is a Interreg project that focuses on Skagerrak. So it's from Interreg Öresund Kattegatt Skagerrak, called Blue Bioclimate. That will be starting now on October 1st. And the goal of this project is to generate tools that span both borders between Sweden, Denmark, and Norway in order to create tools for spatial planning and restoration and for invasive species. So those are the 3 focal topics of this project, and it's a collaboration with DTU Aqua and Aarhus University in Denmark, and then us here in Sweden and the Institute of Marine Research in Norway. And the other project is a Horizon Europe, so it's a EU project called MARHAB, or including trophic interactions in marine habitat restoration is what the long name is for that. And the idea here is to incorporate trophic dynamics in habitat management. So on the EU scale, a lot of the protection and restoration efforts come from the Habitat Directive, which focuses a lot on habitat forming species. But a lot of times other organisms, such as top predators like cod and seals, are really important for the health of the habitat forming species, but that is currently not really included in the Habitat Directive. So that's something that this project aims at, is how we can include that into these EU directives, and then hopefully include that also in management. And this is a collaboration between IMR in Norway again, and us, and SLU as well in Sweden, DTU Aqua, but also the University of Algarve in Portugal, and CNRS in France. So there will be postdocs announced for both of these projects in the near future, so keep your eyes open for that. Thank you for listening.